This concludes the doctor committee meeting of three scholars progress has been evaluated. Now it is time to listen to Professor C. Kamanavalli sir. Dr. Santosh Kumar is going to introduce about sir. He is going to speak on evaluating the impact of endophytic bacteria on the bioavailability of contaminants in phytoremediation systems. Sir has been working on bioremediation using microorganisms and as well as nanotechnology for the past 30 years. Sir is going to be introduced by Dr. Santosh Kumar. I request Dr. Santosh Kumar to take it.
Thank you, sir. Good morning to all. As you know, so yesterday we celebrated National Science Day. On occasion of National Science Day, we are conducting a special lecture program in association with Society of Biological Science and Biochemistry Department Alumni Association. It's my pleasure to introduce today's guest, today's speaker, Professor C. M. Kamanavalli sir, Senior Professor, Department of Biochemistry, Karnatak University, Dharwad. So sir has completed 49 years of teaching experience, having 59 years of teaching experience and more than 30 years of research experience. So sir has completed MSc and PhD from Karnatak University, Dharwad, Department of Biochemistry, and sir started working as an assistant professor, University of Agricultural Science, Dharwad, in 1996. Then sir moved to Karnatak University Science College in 1997 as a lecturer and worked as a senior lecturer, associate professor, and in 2013, sir joined as an associate professor in Karnatak University in Dharwad, Department of Biochemistry, and sir professor, and presently sir working as a senior professor. Having 29 years of teaching experience, 30 years of research experience. So the area of specialization of research is bioavailability, biodegradation, phytoremediation, natural products, and nanobiotechnology and cancer biology. So sir having more than 35 publications in national and international recognized journals, and also contributed 3 chapters. And sir also served as chairman for Department of Biochemistry and also worked as BOA chairman, BOE chairman, and also member for other university, the Board of Studies, BOE and BOE member for other universities. So today, sir is delivering a special lecture on evaluating the impact of endophytic bacteria on bioavailability of contaminants in phytoremediation system. With this brief introduction, I welcome once again for the presentation. Thank you, sir.
Thank you, Professor Kumar sir, Senior Professor and Chair of the Department, and Dr. Santosh. Today I am going to present what I have done little work in during my PhD work and also along with my students' work. Evaluating the impact of endophytic bacteria on the bioavailability of contaminants in phytoremediation systems. Most of the students have studied already in the basic itself, even 6th standard or there is one pollution. Right? The pollution is nothing but the contaminants introduced into the natural systems or ecosystem, natural ecosystem that cause harm to the living beings. That's simple. If you go for your mobile play definition of this pollution, simple way, wrong substances, wrong concentrations, present in wrong places. Wrong substances, wrong concentrations, present in the wrong places. That is what we call that simple. That is just like a mobile definition of this. Power. Phytoremediation. Using a living plant that will remediate the polluted water or soil pollution. That is, the phytoremediation involves using the plants to remove, degrade or contain pollutants from soils, sludges, sediments, surface water and groundwater. Right? These are the materials it can remove some extent, not 100%. I'm not sure that 100%. Then some extent that will remove the pollutants. That is, the pollutant is that is non-biodegradable material that is harmful to the living systems. If you are looking at this picture, this is the picture. This picture, the tree roots can take up the water and contamination from the ground. This is the water table. This water table that's containing lot of the dissolved or undissolved or suspended material that's present in the water itself along with the soil. Or some extent, after some times, that is deposited past, that is deposits. That's why that is contaminated soil, this one is, and water table is also that is in this way. Based on the that is rock level and soil level in the underground, that's underground water. Then suppose if you take a clean, this one is, if you normally the ecosystem some extent that is clean is there, that is clean, that is soil and water is also there sometimes. The water enter through this one is, other this one is, the plants, they, with the capillary force, all the water material will take up and minerals also, dissolved minerals also goes for the, used for the, that is growth of and maintenance of the plants. Right? It is sustainable method. That is a sustainable method to remove this one is continuously. That is not only one time or physical or chemical methods. Physical and chemical method drastic method. That is easy to remove and faster. But this is sustainable. That is natural process of plants to stabilize or degrade or remove contaminants. What are the types of phytoremediation? This one is. The one is phytoextraction, phytostabilization, phytodegradation, rhizofiltration, photovoltaization, and phytostimulation. These are some process that comes under the phytoremediation process. Then simple all the process, that is whatever the process are takes place in the phytoremediation, that is including phytodegradation, phytovoltaization, phytoextraction, phytostimulation, phytofiltration, phytostabilization. All these are that especially takes in the same plant. Right? That's combined. Then Then there are several plants, hyperactivated plant. That is high level of contaminants that is present in the tissues, stored in the tissues. That's why these are special plants have ability to absorb and concentrate high levels of heavy metals from the soil in their tissues. Not only there is heavy metals, nothing but nickel is there, cadmium is there, lead is there, mercury is there. That is so many inorganic materials are. These inorganic materials, sometimes some specific plants we take up and store in the tissues. That is present in the tissue. Those plants what we call that is hyperaccumulator plants. They can take up the metals in like nickel, zinc, cadmium. These are some examples which are typically toxic to most plants. There is certain level. There is already this one is macro micronutrients. The macro micronutrients depends upon the concentration. If a higher level of concentration is there, that is toxic to that is everything. If they are living things, are there microbes, are plants, are humans, or even any animals. That is some extent. Right? Then they can be used for the phytoremediation, which is the process of using plants to clean up contaminated soils and water. Some extent they have to absorb and degrade it, right? Or remove it. Those we can use for using the soil and water. By planting hyperaccumulator plant in polluted areas, especially in the industry, that is industrial areas, they discharge lot of chemicals, either inorganic form or organic form. These are that is ultimately enters into rivers, water tanks, and once again percolated into the groundwater level. This groundwater is enters, then once again extracted for drinking purpose, especially in the what we are using the Tumbabra river water, that is a good, no problem at all. Some extent there is also containing lot of material there, but those villages, some villages, they are extracting the water from the underground. Those people are suffering from such type of that is especially in the dissolved contaminants, that's either inorganic or organic. If you go for the that is Mundagi, Sidarati, etc., there is lot of arsenic content. This lot of arsenic content, sometimes these simple alcohol, these are hinges just like folding and extension. But these are sometimes cuts because of that deposition of that is pollutant material in the joints. Right? Simple, that is presentation of this one is the hyperaccumulator plant, and both are same of course. Accumulation of the metals in the leaves. These leaves, that is after there is some degradation or oxidation, reduction, hydrolysis, etc. Whatever the chemical process is there, all chemical processes takes place because of the plant material containing lot of that is natural products. This natural product, because of large number of functional groups are there. What is functional group? The functional groups are reactive part of the compound. If at all any reactions are chemical reactions, through that functional group takes place because of this natural products contain lot of material, that's all the reactions are takes place and remove some extent, that is used for the that is evaporated or some gas also there. Those also. Then once you absorb this one is the heavy metals or other things, contaminants, organic content, that is transplanted into source. Then these sources are that's once again harvested and destroyed, or we have to extract it from the useful chemicals for your various purposes. The phytoaccumulation. The phytoextraction involves the plant absorbing contaminants. The plants absorbing the contaminants because of already known this one is the various functional groups and enzymes and other biomaterials to absorb the that contaminant material through various processes, through their roots and transporting them into above ground level parts, stems or leaves. This is particularly effective for the process typically involves uptake, that is roots of absorb contaminants from the soil and water because of the transportation, that is with capillary force of the action, dissolved contaminants can be extracted this one is. Then translocation to transported into various roots and leaves, etc. Then accumulation, the contaminants accumulated in the capillary ground level, that is either in the stems or in the tissues or in the leaves, which can be harvested and disposed safely. Then if you are looking at this phytostabilization, the phytostabilization means the focus on the that is reducing the mobility. That is will not be absorbing this one is directly. Restrict your mobility of the minerals or contaminants through in the plant system. That's why the phytostabilization focus on the reducing the mobility of contaminants. either organic or inorganic, and bioavailability for contaminants in the environment, that is root development. That is root development means the extensive roots are developed. I think possibly those who are studying the botany, that is morphology. The roots are various types of roots are there, modified roots are. For example, rhizomes are there. These rhizomes are, that is along with the plant water surface level of the soil, that is extended. Simple example I give to you, ginger. Ginger is also is modified roots, that's all. That is only along with the surface of the soil, that is extended, grown further, including Dharwad also. See, right? The extensive root systems that prevent the soil erosion. Because of that is soil erosion, that is the transportation of material or minerals, etc. That is inert manure or manure materials. That is, if heavy rains are there, all the materials are washed up and floated into get into the that's river. That will be prevented by this type of root development. Then another one is sequestration. Bind the contaminants in the root zone. That is only binding. This one is. That is whatever the contaminants are present in the soil or water that binds to the root, then reducing their leaching into the groundwater. That once you burn this one is the concentration will be goes down in the particular water. That once again restricted the that's contamination of the water. And chemical alterations alter the soil pH. That is soil pH means I think probably everyone you know that what is the irrigation water pH? What is the irrigation water pH? Normally above 10. Because of that is hydroxides. That soil containing lot of minerals are there. That is hydroxide, magnesium hydroxides are there, barium hydroxides are there, potassium hydroxides are there, sodium hydroxide. That is because of that is alkaline nature. The irrigated water usually normally that is alkaline nature. Right? Then the alternative pH of this soil and water redox reactions. That is redox reaction is because of all these reactions, that is hydroxyl groups are there, some other groups are there. That is oxidation reduction reactions are simultaneously takes place in the soil itself without knowing. We don't know any reaction mechanism in the soil, but the microbes and all that are taking place of this one is that is redox reaction conditions precipitating contaminants and rendering them less available for the that is harmful, especially in the to the living beings. Then photodegradation. That is degradation is the whatever the compound is there, the larger compound converted into smaller compounds, or sometimes toxic compounds into less toxic, not 100% toxic, not very good, less toxic compared to this one is. Right? This same. Right? Photodegradation also known as phytotransformation. That is larger fragment into smaller fragments. These fragments are sometimes it may be less toxic. Involves the breakdown of organic contaminants directly by the plant itself because of the plant material containing lot of the enzymes, functional groups, that is reactions, phytoreactions are also take place. That's why this is takes place by plant associated enzymes also. Right? The enzymatic breakdown. Plants produce many enzymes. For simple example, that is dehalogenase. Dehalogenase means either you have to remove chlorine, bromine, iodine, metastine, etc. These are sometimes, if you high chlorine content, that is also harmful to our human being. That's dehalogenase. And nitrogen already you know the nitrogen lot of there is microbes are produced in the there is especially nitrogen fixing organisms. Klebsiella, pneumonia, nitrobacter, acetobacter, etc. These are that is containing the nitrogen enzyme that break down the that is complex organic compounds into simpler and less toxic molecules. Then plant metabolism, the contaminants are taken up by the plant and metabolized into non-toxic or less toxic compounds within the plant tissue itself. Because of there is already some chemical reactions are biochemical reactions are taking place. That's why reduce the toxicity of this one is that may be one of the reasons to less toxic of this organic pollutants. Then rhizofiltration. Rhizofiltration already know this one is this is the plant material that is essential along the surface of the soil. That is already spread, those who are studying the botany, that is Rixia, Marcacea, good bioavailability are this. That is firmly attached to this one is callus systems. That's why particles will not be able to remove that easily. Because of that is large extension, just like a plant where extended the plant systems, especially bryophytes. This one is the good example for rhizome. This one is Markansia, etc. Right? The involves using plant roots, absorb, concentrate, and precipitate the contaminants from the aqueous environment. This once you prevent this one, that is bad-like formations of the rhizome, that will not be easily dissolved in the water because it will not be erosion. That is, erosion will be prevented by this one because of the only thing is the extension of roots that carries in the systems. The absorption, roots take up the contaminants from the water. Precipitation, roots to associate with the microbes, precipitate metals, making them into less soluble. That is, already there is whatever being there is bacteria. The soil is also containing large number of bacteria. The plants also containing large number of bacteria. That is root nodules or roots or etc. That is what you call that is endophytic bacteria. Right? The concentration. The contaminants are concentrated within the root biomass which can be harvested. Photovoltaics. The some of the compounds are that is volatile nature. This volatile nature, sometimes larger fragments convert into smaller fragments. That may be that is volatile nature. That's also we are going to provide with the help of plants through that is stomata. Right? The photovoltaics involves plant taking up volatile contaminants and releasing them into atmosphere in a less harmful form. The one is roots absorb volatile contaminants from the soil or water and transported it into the tissues, stems, etc., aerial parts of the plant. Then volatilization contaminants are volatile and released through a stomata as a gases. The phytostimulation. The some of the organisms are plants. Right? If you plants are released, this one is simple example. The root exudes. Plants release the organic compounds through their roots, such as sugars. The sugars are very much required for the growth of the fungus, especially fungal organisms. The amino acids required for the especially in the microbes, that is including organisms, that is bacteria, soil organisms, fungus, etc. Then enzymes which stimulate the microbial growth and activity. That is, these are the food material available. The large number of bacteria or microorganisms grown on the soil itself. That is to use for the betterment purposes. If you use this one is along with this one is along with this contaminants, they use for the growth and maintenance of these organisms. The microbial degradation enhances the microbial population in the rhizosphere. That is, large number of the microorganisms are that is evolved in the organ that will be modified genetically because of the toxic contaminants are present in the soil. Right? Degrade organic contaminants more effectively than plant development. Then, if you look into the broader way of this phytoremediation, then what are the advantages of that is the phytoremediation of this one? The one is eco-friendly. It will not be harmful for any living beings, either human being or animal system or mammalian or plant or microbes. Right? Natural processes minimizing the environmental disruption. It will not be harmful for the, that is especially in the natural processes. Then cost effective. It will not be much, it will not be cost for this one is expenditure to reduce the pollution of the, especially in the soil and water. That is cheaper than conventional remediation process. Then aesthetic process, if a greenery is more, the visual appearance is also looking into betterment. That is best. That is visual appearance. That is what we call that is aesthetic appeal, enhance the visual appeal of the contaminated site. Then soil health. The soil health, suppose if you remove this, all these things, the nutritional green manure is increased. Green manure means that is manure that is containing lot of that is organic compounds including inorganic, that is including micro, macro, nutrients, etc. That is required for the maintain the soil health. Also required for the microbe. That is good microbes for the growth and maintenance. Then biodiversity. If a greenery is more, the diversity is also biodiversity is also increased. That's one is the encourage the diverse of range of that is plant and animals life. There is various kinds of that is even including the good plant, medicinal plants, only greenery plant, right? Vegetable, natural vegetables will be increased. If natural vegetable increases, then automatically the other animals can also living in the good life or leading in a good life. Then solar power, that is to trap the solar power, that is plants can use their photosynthetic activities more because of the more amount of carbon dioxide using and convert into useful material for the food material. That is the solar power of this one is plants sunlight for growth and making the process sustainable. Opposite to this one is, what are the disadvantages of this one? Disadvantages, the one is time, that is first one is everyone respect for the time. That is time consuming. That is remediation can take several years to achieve significant results. That is not tomorrow's or immediately today's. That is long years to require for this one is longer duration because of slowly it is develop this method. Then depth limitation, not deeper root systems, that is certain level. That is 1 feet, 2 feet, 3 feet, 4 feet, like that. Not too deeper of the systems. One is depth limitation is there. This one is effective mainly for the contaminants within the root zone. Right? Not suitable for the deep soil contamination. Then contaminant specificity. There is certain specific plants are there. These specific plants only specific floatants only absorb or binding. The rest of the contaminants will not be binding. This one is, that is what we call that is specific contaminant. Different plants target for the different contaminants require specific species for the specific pollutants. Either metal or heavy metals or macronutrient or organic material or polycyclic aromatic hydrocarbons, etc. Right? Then plant toxicity. High level of contaminants can be toxic to plants. That is even the sugar is to some extent is good to taste for ourselves also. If too much of sugar is will not be eaten, then automatically toxic to our bodies. The same process here. High levels of contaminants can be toxic to plants using the remediation. Then contaminant release. That is after there is hyperaccumulating plant or specific plants we have to extract this one is have to safely we have to dispose of. That is another kind of that is method you have to use for the removal of this one. How you are going to remove this one? Suppose if you take it as plastic, that is plastic is also non-biodegradable material. That is always working for me, that is your gutters, some channels, water channels, logging for this one is. Then safety material is very important. You segregate this one is and put it in one bag. This bag you have to use for the making roads and all that, including that is mixture of that is tar, coal tar. Alright? That is harvested plant biomass containing contaminants must be disposed of safely to prevent re-release into the environment. That is once again, that is another kind of circus we have to do in this place. Then effective climatic conditions. Effectiveness can be influenced by local climate and soil conditions. If the soil conditions is good, the contaminants will not be entered into our food chain. If we enter the food chain, there is contaminants, then automatically what we are going to do? We have to use the food items are consuming, then once again re-enter in the system. That's avoid that. Some examples are quoted here. The mechanisms of this one is the Indian mustard oil, sashimi, right? Then specifically, the phytoextraction Indian mustard plants absorb the lead from contaminated soil. That plant absorbing contaminant. That is only lead. Because of lot of lead will be introduced into the ecosystem because of mixture of the petrol. That's why nowadays they are mixing the blend petrol. Blend petrol means there is 25% of petrol, 75% of ethanol, or 50% of ethanol, 50% of petrol, and that is 75% and 25%. That is what we call that. Today, what we are using that is blend petrol. Because of only thing is reduce the one is import of the that is oils from the Gulf countries. The second one is to reduce the pollution of the lead concentration increase in the soil or environment. That will be that once again main motor is blending of the petrol also. Right? The other one is willow tree. That is specifically that is willow tree stabilizes the soil contaminated with the arsenic. If you go to the northern side of the that is especially in the along with the Tumbatra river, that is after hygienic. After hygienic, there is the blood containing lot of arsenic. That arsenic it causes lot of things up. That is teeth becomes that is decaying is there. Bones become degeneration is there. That is stunted growth is also there. Will not be folding properly. Right? These are the some disadvantages of the that's arsenic. That's why there are certain plants you have to grow in a high contaminated area. Can some extent you have to reduce the pollution of that is arsenic content. Then photovoltaics. Please clearly you have to use for the selenium from soil. That is such type of plant you can go for the that's just like an ornamental plant. This one is right. You have to use reduce the selenium concentration. Sunflower. I don't know to realize that is oil. What we are consuming this one is oil for cooking it. It is not a natural one. You have to think of first. This is not a natural one. That is synthetic one. Purely. That is extracted from that is oil material. Because of the 1 kg of that is sunflower seeds. That is, I think probably 350. Just you have to think of 3 to 5 kilos of that is seeds. It is to generate 1 liter of oil. How much will oil cost? 150 at the most. That to genuine. That to genuine sunflower oil. Yeah, what you are using cooking. That is 150 rupees or 200. That is what is the cost of this one is? That is more than 2,000 nearly. Whether company people are there thinking of this one is? No. Naturally, they are going for the, this is not a natural oil what we are consuming today. That is synthetic one. Right? That is generated from the that's petroleum products, etc. Hydrocarbons. That is reprocessing there, of course. That is, this one is specifically absorbed, that is uranium. I think you know the uranium nature and all that. Those who are studied in the earlier class chemistry. Then notably, that is phytoremediation projects in India. Just simple. The one is Loktak Lake, Manipur. Loktak Lake, Manipur, Ramsar site is in Manipur has been undergoing phytoremediation efforts. The lake features floating islands called humid, which play a significant role in the phytosanitization, bioavailability, and absorbing sewage and agrochemicals. Because of, if you go to the rightful side, I think hopefully Santosh is better than me. You know, that is whatever the rice we are eating. This one is there are lot of pesticides they are using for this. Lot of even suggestion of the agriculture scientists they are suggesting that is 10 ml or 20 ml of the pesticides you have to spray for the to prevent the disease of the paddies. That mixing of that's 120 ml, 100 ml, 200 ml, etc. Drastically, they are thinking of higher amount of that pesticide if we use for this one is immediately control the disease. No, that's not a possibility of this one. Because the microbes or insects can resist this one. That's such a level of that is resistance of this pesticide. That's once again disadvantage for this one. Then Herbal Lake Bangalore. Herbal Lake Bangalore in suburban Bangalore was successfully disseminated through bioavailability. The project involved in the use of artificial fruiting wetlands with a select plant species, mechanical irrigators. That is mechanical irrigation means that's whatever that's supposed to be water is stagnated water. The lot of large number of toxic organisms will be generated or grown. If the aeration is there, to some extent can be not be grown for the some organism. That's why the aeration is required for this one. I think hopefully those who are traveling from the Bangalore to Tumkur, in between some rotors are there in the stagnant water. I think hopefully everyone have to see it. Then improve the impact of the combustion and issue. Plants are used to stabilize the extract and heavy metals from the ash. If you look into the ash, whatever we generate, we are using the power. That's electric power. There is a lot of power will be generated. That is less amount of power is generated from the Sharavati hydro project, but lot of power will be generated in the especially in the Raichur Shakti Nagar. I think probably the goods are not to travel from your Dharwad. That's especially they via from north to via that's Goa to Hubli Dharwad and reach to Raichur. That is n number of tons will be fly ash produced from the coal. After the burning of the coal, once it generated this one is the ash problem is avoided. There is so much of there is complications are there. This ash nowadays they are using for the one project when I was in the agricultural university. Fly ash utilization in the agriculture. They are using for the agriculture purpose, but the mineral content is very less because already burned that. Only that is high melting point. High melting point, the minerals are means the rest of the lower melting point components are already burned that one is. But that will not be proper use for that is fly ash because the minerals are very less compared to this one. Only thing is that to some extent may increase the macronutrient. That's all. Not that one is. Then another thing is the another disadvantage. After using this one is generally the fly ash. That is, if you mix and comes out, that's all the density is very less of these particles that is deposited on the plants. And also the air consuming those peoples, their fate is also that is right? That's one. Then these are the some experiment what we have done in our laboratory. Right? Experimental studies on the phytoremediation. The one of my students is garlic and cilantro. They are using the mixing, they extract the garlic extract and cilantro is what we call that kothambari. Kothambari. They extract this one is extract and mix it with the heavy metals and irrigate it as spikes to the plants. The plants, although zinc metals or nickel metals or copper etc., that is lesser amount of that can be absorbed and transplanted into various tissues. When you are mixing with the garlic or cilantro extract, the higher rate of that is absorption being more in the plant because of there is some complex form along with this one. Right? That's why the garlic and cilantro extract better extraction of the zinc using sensitive arthropogana. This sensitive arthropogana is a what you call that is in commonly snake plant, your ornamental plant. I think probably some of the people there cultivate this plant in the ornament purpose. Snake plant, this one is sensitive arthropogana from the contaminated soil. These are the normal soil that is isolated from this one is, and this one is the phytochemicals present in the soil. You see this one is the enhanced zinc extraction ability of the Sansevieria roxburghina plants with the natural chelates. The natural chelates already we are using, garlic also we have used excessive, and cilantro is also using for this one is for the cooking purpose. But if these mixtures are mixed with the metal solutions, it will not be harmful in nature, it will not be affected in any ecosystems, natural ecosystem. Right? This is the control. If you take it, that is 400 milligram of this one is, that is, this is the control. Plant with zinc. This is the plants that is spreading with the zinc solutions, only zinc solutions. Alright? Then lesser extraction should be there. If you mixing with this plant with the zinc and garlic extract, that is once again the zinc extraction is more. That is absorption is more. Because of that is some functional groups reacting with this natural systems, and along with the absorption is also more and translocated is because of natural. Both are plant originated. Similarly, if we concentration, zinc concentration increase to that, the initial concentration increase 400 to 600, still more absorption is there. Right? Using natural one. This one is. Right? If you go to the soot system, once again you have to estimate how much amount of zinc is that is extracted or transported or translocated in this one is. That's also you have to compare to this one is huge amount of the zinc is extracted. Then similarly, the treatments of the soil that is remaining, that's how much amount of soil containing that is remaining, unutilized, that's something I can by this one. That's why the concentration of zinc with chelators, that is root, shoot, residual soil of the sensory transformation, that you have to estimate by this one. Then how that is one is bioconcentration factor, BCF, simple form. The initial constant bioaccumulation, that is how much amount of that is particular metal accumulated in the plant tissues, that we have to extract. Right? Process can be measured by using a bioconcentration factor. BCF is the ratio of metal concentration in the plant tissues, its concentration in the metal, that is soil and water. Once again, that is calculated. That is bioconcentration factor equal to, that is C is the concentration of ornamental plant, that is concentration. Then remaining, that is residual, the metal concentration remaining, that is SL. That is concentration of the that soil remaining unutilized or unabsorbed. That's also you have to calculate. If it, this one is, that's once again this is the water, that is control water. The integrated soil containing The one is without only any cilantro or garlic extract. The one lot is that cilantro with zinc. The another one is garlic and zinc. The another one simple zinc. If a comparison of this one is, that's once again garlic and cilantro extraction is more of the zinc better than this one. When I am mixing with natural product, will not be harmful to this one. Then similarly translocation factor. How much amount of translocation from root to that shoot? That's what we have to calculate for this one is. That is translocation factor equal to the concentration of the shoot sample. That is how much amount of that is heavy metals in there. Then similarly concentration of the root remains in the root. That is to be translocated later. That's what we can calculate. The phyto extraction, that is total how much amount of that is, what is the phyto extraction? That is time dependent. That is 24 hours, 48 hours, 72 hours, 1 week, 2 weeks, etc. Because this one is, that's once again you have to calculate, that's phyto extraction rate of this one is, that is equal to percentage, that is metal of the ornamental plant, metal of the, that is soil, that is remaining residual into multiplied by this one. Then, if you are looking into the, that is instrumental analysis of this one is, that is qualitative analysis different. I think mostly you have done in the base level, qualitative, chemical, qualitative analysis of all that is inorganic metals. Right? The acid radical, basic radicals, or positive radicals, or negative radicals, etc. Right? If you take this TG, thermal gravimetric analysis of this one, the thermal gravimetric analysis means without any metal, there is single stage decomposition. If you see this one. This is the single stage decomposition. If you concentrate on the metal, it is increased. That once again, up to certain temperature from here to here, there is one stage is there. There is certain elements or certain, there is low melting point components will be, there is degraded, decomposed. Then if the temperature is increased, there is still more, that is higher temperature wasted metals can be removed. That is to form converted into gas. If you go for the TCA example. Then DSA, that is whether the chemical reaction is exothermic, endothermic, that we calculate using this one is, that is based on the enthalpy. Right? Here, the added zinc and natural chelates significantly change the enthalpy values. Suppose if we take a zinc concentration is 600 milligram per liter, that is along with this one is garlic, GL is garlic. That is enthalpy is 28.6. That's here. Right? If you go for the same one is 600 cilantro, that is endothermic reaction, that is endothermic reaction, that is endothermic reaction, exothermic reactions will be better than me because we already learned it earlier as well as in the MSc course because either heat absorbed or heat reactions. Right? This one is this page. Then simple experiment we have conducted in our laboratory. Then one is water, that is what you call, that is Pistia stratigraphy. I think probably everyone you are seeing in the lakes normally floating. That is very good appearance of this one is if you looking into this one. You can cultivate one your home also. That's simple. Right? This one is this is the plant. Pistia stratigraphy. The plants we cultivated in the water itself. There is little amount of, that is normal soil we have to mix and water. That is water floating plant. This is the normal. Then where zinc concentration or any metal concentration is added to this one is, you see the situation of the plant there. That is, it may be death also occurs, it may be breaking of lips, etc. Right? If you look, go to the copper, that is copper, we have conducted the copper, that is soil containing, that is water containing copper, we have to introduce this one is. That is 0.5, this one is bioconcentration factor, that is this one is translocation factor, this one. Suppose if you go for this one is higher concentration 1.5 mg per liter of the copper, this one is the same increase the concentration, that once again touch this one. You see this one. As and when increase the initial concentration of this one is the simple effect of initial concentration of the Pistia stratigraphy plant. Go on increasing when there is metal concentration, the plant will not be survived. That means simple toxic to plant. That's right. See this one. Another one is the treated copper solution. Suppose water, this one is normal water. shoot sample and root sample you have to extract it and see this calculation of the how much amount of carbon present in the roots, shoot and leaves. That also you have to calculate. If you're looking at the chlorophyll content, you have to estimate this one. The total chlorophyll content, etc. Chlorophyll A and B. If you're looking into the chlorophyll concentration of this one is the total concentration. This one is the green one. Right? When there is higher amount of toxic materials present in the soil or water, then reduce the chlorophyll content is also. Right? This one is see the same one is. Right? The concentration of the concert is increased. This is the situation of the pond. The pisceous structures are used. We have taken that we will go for the test once again thermographic analysis. This is also DSC. Then another one is already asking this one is that is alluvial plant. That is endophytic bacteria. The alluvial plant containing lot of endophytic, that's good bacteria. This is this one. This is the normal plant. Then this is the control media. There is no microbes at all. The same roots you have to put it in here. Stabilized roots placed on the other place. Then you have to see this one is bacterial growth from the roots around this one is. See this one is. See that is quite best, sir. You compare to this one is. Then after this one is, if you go for the subculture of this one is, these many number of that microorganisms present in the root. That is what you call that endophytes, endophytic bacteria. And then once again, the microscopic images, then once again harvested, you have to go for the biochemical analysis of this one is, that is morphological and biochemical characterization of the isolated organism. Then among n number of organisms are developed, only one organism will be identified through NCS. Right? Then this is the appearance of the endophytic bacteria. Right? And they extracted the genomic DNA. This one is that is more in the NCBS because they are carried out this one is. You see this one, genomic DNA of this one. That's compared to this is the standard ladder in the ladder. This one is isolated from the that is isolated microorganisms. This is atomic force microscope. The how extent that is length of the bacterium that we have to measure here. And this is one is the bioavailability of this one is. And this one is, if you go for the scavenging activity of this one is, that is depends upon the chlorophyll content and contaminants present in this one is, the scavenging activity also goes down. And the ion spectra of the, there are certain, the reds also you have the dyes you have to use, one is carbonet. This also, that one is degraded carbonet. Bromophenol blue, this one is, that's also dye. And this is degraded bromophenol. That is after degradation, you have to put it in the water and run the microorganism, then reduce the toxicity of this one is, then goes on, that is concentration also increases, then modify this one. This peak will not be appear in this one after degradation of this one. You see this one. Right? If it, this is also, that is concentration is less. Then some other, the compounds may be generated, that is what you call that is intermediate compounds. This intermediate sometimes can be identified or unidentified because of large products are formed, especially in the natural system. Very difficult to identify the product because standards are also required. Only thing is, certain extent we have to analyze it using HPLC and mass spectra only. This is the HPLC analysis. Right? This one that is polyaromatic hydrocarbons. That is more than one, that is benzene. Polyaromatic hydrocarbon. One is anthracene, another one is phenanthrene, and these two mixing, that's what we have to try. That's how much amount of that is the bioavailability hydrocarbons replaced by these microorganisms and reduce the plant cancer by the That's how we have to. These are the HPLC, GC, etc. That is mass spectra. And finally, octadecanoic acid is the product of this one. There are some actual, what is the degradation of foods? That is converted into, that is phenanthrene converted into this one is, this compound once again further degradation, green closure, that is ring purpose of Green cleavage occurs, green closure often that is performed with target and enters into TCA cycle. Right? That is, thus all the products will not be used for the same organism. There is different organisms, there is different compounds, may be used for this one. Will not be a same amount of sugar will not be same sweet will not be accepted for everyone. One is accepting X, another one accepting for the Y, another person X interest for the Z, etc. The same way here is also. The microorganisms very specifically have to use for the particular compound as a growth substrate. Right? Now I get to some other product. Right? Simple example other things like considering present in one species, the isomeric compounds. The isomeric compounds can be separated by that penicillin species. One particular component at D or F can be used for the separation of this using the fungal species also can separation. This is the that is anthracene degradation roots of this one is finally there is 6 active acid is the end product of this are the intermediates. The summary of this one is the added chelating agents that one is cilantro garlic extract modify the presence of the bioavailability of the heavy metals in the contaminated soil for the increased absorption of the zinc by plant. Garlic extract was able to significantly enhance the zinc uptake, that is because of natural product by plants with highest translocation factor and bioconcentration factor in the plant. Then pisceous roots simple significantly improve the copper absorption with the chlorophyll depletion is also great data also. Then presence of the work gather a necessary knowledge of the design biotechnological tool for the that is AM1, that is isolated organism and degrade that is anthracene, phenanthrene, conglomerate, monosulfide, etc. Right? Microbes assisted phytoremediation of Enadium spiker that is gradually ministry of plant endeavoring the that make them first move towards the producing the gradual changes in the that is led to an improvement of the agrosystem. That is once again the agrochemicals what we are using that maybe some extent we can reduce the pollution of this one. If you reduce this one is then once again leaching of the that's environmental pollutants into the soil and water that may be some extent you have to prevent because otherwise that's heavy metals or heavy agrochemicals enters into the food system that once again will not be able to cope up with those concentration of the substrates. Then if you use it that is FTR, GCMS spectrum, phytodegradable unaromatic hydrocarbons in soil confirm the presence of phthalate, octanoic acid as a major intermediate in the phthalate part. Right? Ring opening and re-element reactions are so many reactions are taking place without knowing. That is because of there's a wonderful biochemical reactions are taking place in the either the human metabolism or in the plant metabolism or in the microbial metabolism. Right? The phytodegradation method may therefore promising approach for the remediation of especially in the aromatic hydrocarbons. Right? I thank finish up this one is. He is now is appointed as a cadet officer in training, Nagpur. His wife is, she is almost all work, phytoremediation work carried out by, she is Manisha Shankar. And she is also doing now, it is working on nanotechnology. She is representing the states, no? And he is also qualified for the net and she is a set net, gate, etc. All this is also. And these are the processes we are working on the bimetallic compounds, nanoparticles. He is also working on the, there's some techno-medicinal plants on this one is, and he is also about the company, the nanotechnology. Some more people are there, are possible.
Now it's open for discussion. Do you have any questions?
If at all any questions are, the first question.
You can ask any questions. Have any doubts?
I think probably good plant biochemistry sir is there, good microbiology is there, no doubt at all.
Question? seniors plant biochemistry ಯಾರು ಓದ್ತಿದ್ರ? Metal stress is ectopic artery, right? Okay, there are phytochelatins and metallocyanins, two types of proteins that can bind to the metals. Napoleon Bonaparte, the famous ruler of France, was killed due to arsenic poison, one reason. Another reason is mercury poisoning. And as Sir said, arsenic, you cannot move the hands freely and department the joints. In biochemistry, we need to look at which enzyme is inhibited by arsenic. The first thing is that pyruvate dehydrogenase of citric acid cycle, and before citric acid cycle, that enzyme converts pyruvate molecule into acetyl-CoA. That is the step which is inhibited by arsenic. In addition to this one, even enolase, glycolytic enzyme, that is partially inhibited by arsenic. And Bangladesh, which is well known for the arsenic contamination. And I am surprised to know that Sir told about Mundargi, then near Raichur, and the nearby regions of Tungabhadra. And to see now how arsenic poisoning it is affecting the people over there. And recent news says selenium if it is increased, that leads to baldness. So there were, especially in hilly regions of Punjab and Haryana where wheat was grown, that wheat grains were provided to the people in Maharashtra through public procurement system, that is ration shop. Through ration they have served the wheat, and people consumed that wheat in a village. The whole village, male population especially, they were affected by hair loss. So all they have become bald. And one of the person who is having some scientific interest, he conducted some study and he could able to find that huge amounts of selenium deposition is accumulation in the wheat grains. So that is the reason for it. In fact, selenium is said to be one of the antioxidant molecules, metal ions, and this selenium is used as antifungal substance in shampoos which are used for the treatment of dandruff. Now we must be very cautious that selenium included shampoos and as well as hair applications, whatever oils or soap if that are available, that must be carefully used. So rather than getting protected from fungal infection, we may be having a threat of losing hair, right? So there are many things. Being biochemists, we should have always, our perception should be like which enzyme, which metabolic pathway, what could be the reasons, what are the organs that are affected, all these things. So it is the curiosity we need to develop. On behalf of department and my personal behalf, though there is a formal vote of thanks rendered by Dr. Pratap, I thank profusely Professor C.M. Kamanavalli sir, very good friends, not simply colleagues, for enlightening all of our students and as well as scholars and staff members. It was a wonderful talk on phytoremediation, sir. We have a paper on plant biochemistry in third semester. Some of the concepts are yet to be taught to them. So in next couple of days that the aspects are going to be taught to them. Thank you. Now I request Dr. Pratap to
ಸರ್ ಒಂದೇ ಮಿನಿಟ್ ಈಗ ಇಲ್ಲ ಸರ್ ಇದೇ ಮೇಡಮ್ ಈಗ ರೀಸೆಂಟ್ಲಿ ಈ ನಾರ್ತಕ್ಕೆ ಏನಾಗಿದೆ ಮೊನ್ನೆ ಗುಲ್ಬರ್ಗದ ಒಬ್ಬರು ಮೇಡಮ್ ಅವರು ಡಾಕ್ಟ್ರಿಗೆ ಅವರು ಪದ್ಮಶ್ರೀ ಅವಾರ್ಡ್ ಬಂತು, ನೀವು ನೋಡಿರ್ಬೇಕು. ಅವ್ರು ಹೇಳ್ತಾ ಇದ್ದಾರೆ ನಾರ್ತದಲ್ಲಿ ಹ್ಯೂಜ್ ಅಮೌಂಟ್ ದೇ ಆರ್ ಯೂಸಿಂಗ್ ಪೆಸ್ಟಿಸೈಡ್ಸ್, ಹಾಗಾಗಿ ಎವ್ರಿ ಹೌಸ್ 1 or 2% ದೇ ಆರ್ ಸಫರಿಂಗ್ ಫ್ರಮ್ ದಿ ಕ್ಯಾನ್ಸರ್. ಅಲ್ಲಿ ಒಂದು ಈಗ ನಮ್ಮಲ್ಲಿ ಹಂಗೆ ಕ್ಯಾನ್ಸರ್ ಪೇಷೆಂಟಿಗೆ ಹಂಗೆ ಕನ್ಸಿಸ್ನಲ್ ರೇಟ್ ಮೇಲೆ ಕೊಡ್ತಾರೆ ನಮಗೆ ಟಿಕೆಟ್ ಹಂಗಲ್ಲಿ ಕ್ಯಾನ್ಸರ್ ಟ್ರೈನ್ ಅಂತ ಐತಂತ ಯುಪಿ ಒಳಗೆ ಕ್ಯಾನ್ಸರ್ ಟ್ರೈನ್ ಅಂತ ಹೋಗ್ತದಂತ ಒಂದು ವಿಚಾರ ಮಾಡಿದ್ವಿ. ಅಂದ್ರೆ ಎಷ್ಟು ಜನ ಇರಬೇಕಲ್ಲಿ ಅಂತ ಆ ಟ್ರೈನ್ ಒಂದು ಆ ಟ್ರೈನಿಗೆ ಹೆಸರಿಟ್ಬಿಟ್ಟಂತ ಕ್ಯಾನ್ಸರ್ ಟ್ರೈನ್ ಅಂತ. Handigodu, that is Mangalore, that is they are using endosulfan, that is also the biggest problem especially in the Kasaragod and Mangalore people, including Udupi, up to Goa Bali. They are suffering from this one.
So what about people are growing vegetables in drainage water, contain heavy metals and compost?
Everyone they are offering for the very fresh one. If a fresh one is very good in there, that's heavy amount of their pesticide they are using. That's why the insects or microbes will not be able to eat those things, but the same thing will be eaten by ourselves.
who are supreme as our
ಈಗ ಯಾವ ಇನ್ಸೆಕ್ಟ್ ತಿನ್ನಲಿಲ್ಲ ಅಂತಂದ್ರೆ ಅಷ್ಟೊಂದು ಅನ್ಲೇಬಲ್ ಅಂತ ಇರೋಂಥದ್ದು ನಾವು ಮನುಷ್ಯರು ಕನ್ಸ್ಯೂಮ್ ಮಾಡ್ತಾ ಇದೀವಿ. ಯಾಕಂದ್ರೆ ನಾವು ವಿ ಆರ್ ಲುಕಿಂಗ್ ಟು ಫ್ರೆಶ್ ಒನ್ ಗುಡ್ ಒನ್. ನಮಗೆ ಯಾವುದು ಹುಳ ಕಾಣಬಾರ್ದು, ಇದಾಗಿರಬಾರ್ದು ಅಂಥದ್ದನ್ನ ನಾವು ಚಲೋದು ಮಾಡ್ತೇವೆ. ಮತ್ತು ಹಂಗೆ ಡ್ರೈನೇಜ್ ಒಳಗೆ ಅದಿರೋದು ಬಿಕಾಸ್ ಲಾಟ್ ಆಫ್ ಥಿಂಗ್ಸ್ ಆರ್ ಎಂಟರ್ಸ್ ಇನ್ ಟು ಸಿಸ್ಟಮ್. ದಟ್ಸ್ ವೈ ಯು ನಾಟ್ ಬಿ ಅಟ್ಯಾಕ್ಡ್ ಬೈ ಎನಿ ಸಿಂಪಲ್ ಡಿಸೀಸ್. ದೆನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಇಸ್ ಆಲ್ಸೋ ಮೋರ್.
Good afternoon, everyone. I extend my deep gratitude to Dr. Kamanavalli sir, Senior Professor, Department of Studies, Biochemistry, Karnatak University, Dharwad, for his presence and delivering a wonderful lecture. Thank you, sir. Now I would like to thank our beloved chairman Dr. Vaddapudi Kumar sir, senior professor of our department. Thank you sir for organizing a wonderful lecture series program. Now I would like to thank Dr. Santosh Kumar sir, assistant professor of our department. Thank you sir. Now I would like to thank all teaching faculty, research scholars and non-teaching staff of our department. Thank you all. Last but not least, I would like to thank all my dear students listening so patiently. Thank you, Anita.